Welcome students to a short lecture on the chemical and physical processes of digestion, which is what the physio-X activity that's associated with this unit is about. So when we talk about physical digestion, we're really talking about breaking the food down into smaller pieces. We're not breaking chemical bonds, we're not releasing any sort of energy, we're literally just tearing the food into pieces. Mastication or chewing is one of the ways that we physically break down food. Segmentation, which occurs in the stomach and small intestine, that kind of mixing and having the kind of pieces of food kind of bounce off each other, that helps physically break down the food. There's an extra layer of muscle in the stomach for just that purpose. The denaturation of proteins that occurs in the stomach as well. The hydrochloric acid from the stomach acid kind of unfolds the proteins out of their kind of shapes. And then emulsification of fats, which is something that we need bile from the liver to do, is physically breaking kind of the fat droplets into smaller fat droplets. All of those types of processes are physical, mechanical breakdown, no bond breaking. Conversely, chemical digestion is actually taking those foods and breaking them down into kind of the constituent parts. We're literally breaking the bonds between the molecules, which releases energy. Now, the process by which we do this is called hydrolysis. It's the addition of a water molecule between the two monomers, and that splits them apart. So we take all of our foodstuffs and we actually break them down into kind of the constituent molecules. We can't do that in the body without enzymes to catalyze the reaction, to speed it up. So you can see here is a reaction without an enzyme. You can see it's got this really, really high activation energy, but with the enzyme, you get less activation energy needed, and so the process can occur more quickly. And pretty much every digestive process in the body requires enzyme catalysts. Now, the enzymes that we happen to use are hydrolases, because this is a hydrolysis process. So you can see here's our enzyme. You can see here is its substrate. So our substrate in this instance is a dipeptide, so two amino acids linked together. Well, those amino acids will bind to the enzyme. The enzyme will change shape and make like kind of the perfect space for this hydrolysis reaction to occur. Water is added, that breaks the bond, releases energy, and then the enzyme returns to its original shape, ready to do its job all over again, while the products of the reaction are released. In this instance, the products would be two amino acids, which could then be absorbed by the small intestine. So that absorptive processes are a little different for carbs, proteins, nucleic acids than they are for lipids. So when we're talking about absorption, we're talking about moving the stuff out of the lumen of the GI tract and actually into the body. Now, carbs, proteins, and nucleic acids, they get absorbed via active transport. We need carrier proteins in order to do that, and then they get diffused into the blood capillaries of the typically small intestines, and then that then blood gets shunted to the liver through the hepatic portal vein, so the liver can process those products of digestion, the carbs, the proteins, and the nucleic acids. Lipids have a slightly different process. Lipids are absorbed via passive diffusion, just directly through the phospholipid bilayer of the intestinal cells, because lipids are fats and phospholipids are fats, and so they're soluble with each other, and so lipids can just be absorbed directly. They also don't go into the blood. They get absorbed into the lacteals, which are these special components of the lymphatic system. And then the fats actually travel with the lymph fluid for a little while until the lymph fluid joins the regular blood circulation. So if we look at the carbs, carbohydrates, carbohydrate digestion starts in the mouth. So the long chains of carbohydrates, things like starch and glycogen, get broken into smaller chains by the salivary amylase. Those smaller chains will then be broken into disaccharides via the pancreatic amylase. And then the disaccharides themselves will get broken into the monosaccharides via the enzymes coming from the brush border. And you have a different enzyme for the different disaccharides. So sucrase is what digests sucrose to glucose and fructose. Lactase does lactose to glucose and galactose, and then maltose does the maltose. And then once we have these monosaccharides, that is what we then actually absorb into the blood: glucose, fructose, galactose. If we look at the proteins, protein digestion starts in the stomach with pepsin. That takes these kind of long chains of amino acids and breaks them into smaller chains of amino acids. And then we get trypsin from the pancreas, and then a bunch of brush border enzymes like carboxypeptidase and aminopeptidase. The pancreatic enzymes tend to break these kind of smaller chains into dipeptides, and then the brush border enzymes break them into their individual amino acids. So very similar to the process that we saw with carbohydrates, but starting in a different point. If we then look at nucleic acids, their digestion is completely within the small intestine. The nucleic acids themselves, the DNA and the RNA, are broken down into the nucleotides via nuclease enzymes from the pancreas, and then the nucleotides themselves are broken into their individual building blocks, the adenine nucleotides, the ribose sugars, etc., via the brush border enzymes. And that's what actually gets them into the kind of individual, like, phosphate ions, bases, etc. And then it's these individual things that will then be absorbed via active transport. If we look at that active transport, you'll see what happens is on the kind of basal side, the side closer to the blood, here are our intestinal cells. We have this sodium-potassium pump, and it's constantly pumping sodium out of the cell into the interstitial fluid of the body. Well, that creates a gradient for sodium between the lumen of the intestines and the cells themselves. So that sodium then diffuses down its gradient and co-transports the sugars, the amino acids, etc., with them. So it's a co-transport process. And then those monosaccharides or amino acids or nucleic acids, the nucleic acid figure would look just like this, but no textbook I've looked at actually has a figure for nucleic acids, but it would look very similar to this process. They are co-transported in with the sodium and then are absorbed by the blood capillaries and then shunted to the liver. Now lipids are a little different. Lipid digestion occurs a little bit in the mouth. You have a tiny bit of what we call lingual lipase, which does some lipid digestion. A little bit in the stomach too, but mostly lipids are digested in the small intestine. And that is because in order to digest lipids properly, they must be emulsified first. Once the lipids are emulsified, then lipase enzymes will break all of these fats actually into the monoglycerides, into the fatty acid chains, and then those will then be absorbed by the body. So emulsification is accomplished by bile, particularly the bile salts in the bile. It's made by the liver, stored in the gallbladder, and essentially what bile salts do is they break big fat droplets into smaller fat droplets. They happen to be what we call amphipathic, meaning they have both a non-polar region which likes the lipids and a polar region which can kind of face out towards the kind of watery lumen fluid. And so when you have bile salts coating this fat droplet, it will essentially just split the fat droplet into smaller fat droplets. Now, emulsification is a physical digestive process. We're not breaking bonds. We're not releasing energy. We're just literally taking a big piece of fat and breaking it into smaller pieces of fat. Once the fats are emulsified, that's what you're seeing here. We get the bile from the liver, big fat droplets turned into smaller fat droplets. Then chemical digestion will take place, lipase, colipase. That's what actually breaks it into these fatty acid chains and monoglycerides, kind of the building blocks of the lipids. But the fluid in the lumen of the intestines is pretty watery. Fats and water don't like each other. And so the newly released building blocks essentially cluster together into a kind of hiding from the water in what we call a micelle. It is the micelle that will kind of transport the fats to the kind of surface of the intestinal cell. The fats inside the micelle basically breaks apart, then the fats are absorbed into the intestinal cell, but inside the intestinal cell is cytoplasm, which is so really watery. So again, the fats will get kind of packaged together into what we call a chylomicron, and then it is ultimately that chylomicron that is absorbed not into the blood capillary, but into the lacteal. And there the chylomicrons, the fats, will travel with the lymph until they are eventually distributed back into the venous circulation. So lipids are a little bit different compared to the carbs, proteins, and nucleic acids. But that's a nice little summary of how physical and chemical digestion takes place in your body. That's how we get all of our energy and building blocks and nutrients from all of the wonderful food that we eat.